So, thank you for having me. My name is Bob Blasdel. I'm the research director for Vizel Pharma in Egezé, Belgium. And I'm going to be talking here about therapeutic phages as APIs for magistral preparations. But for the most part, I'll be largely taking off my corporate hat and just introducing the field in Belgium as it currently exists from a more academic kind of perspective. So I'm going to go quickly through the historical and current context for the model, discuss quickly how the model works in practice, and then discuss a little bit about about the purpose and the value of the model that we see it has. So, very quickly after phage therapy was originally developed by Durrell, he and Eliava developed intestaphage and then piophage as really, I think, the first good model for phage therapy, where in this model, a cocktail of phages was developed against the assemblage of bacteria that was ailing people in the Republic of Georgia. And then that cocktail was updated regularly every six months to every year with new phages without necessarily taking the old phages out, creating a cocktail that got increasingly more complex and more interesting over the course of the 80 years that this has been done, creating something that's really quite beautiful and interesting. But then, now, largely in the West, most companies are working with a cocktail-based model, developing a fixed drug, where the basic idea is to take a collection of phages that will hopefully hit the widest possible array of strains that are of a specific species that are ailing patients either globally or in a specific region. And then this cocktail will eventually need to be updated in largely the same way. And so looking down the road, the idea will then be to create a new cocktail, which will then need to be tested for safety and efficacy all over again. And then again, continuously, as antibiotic resistance will continue to not go away and we will continue to have problems with problematic bacteria. But then how often will these cocktails need to be updated? These cocktails will clearly last less long than antibiotics have, but how much less long? Will they need to be updated every 6 months to every year, like the Eliava cocktails? It's, at the moment, unclear. Whereas from the beginning, there was also always a personalized model for phage therapy, where a specific patient would come into a clinic presenting with a problem caused by a bacteria. That bacteria would be isolated, and then phages would be isolated either specifically against that bacteria or would be tested against that bacteria to determine which phages or isolate phages that would then specifically infect that bacteria for that specific patient. So in the modern era, we see this model making sense as having a large library of annotated, characterized, and manufactured phages that would be available for a companion diagnostic to choose between for patients who would come into a clinic, creating a personalized cocktail for each individual patient based on a phagogram companion diagnostic. So, in contrast between these two major competing models, a phage cocktail is really the object that's being invested in. It's the center of the business model. That cocktail will need to be manufactured GMP. It will then need to be validated in preclinical tests and then validated in phase 1, 2, and 3 trials in order to gain review and approval and marketing authorization. And then, obviously, it will need to be surveilled. But this whole process takes, on average, around 10 years, at least. And there's the promise of, at some point eventually in the not quite foreseeable future, having eventually a rapidly updating and licensing procedure that might be adopted like for influenza vaccines. However, now we have available with current legislation the ability to do the personalized model, where how this would roughly work logistically is a regularly updated bank with quality-controlled small-scale phage products will then be made available to laboratories, which will then select phages against the clinical isolates from individual patients, which can then be formulated into an individual cocktail of phages, which can then be applied to a patient. And then when phage-resistant isolates of bacteria are observed, they can then be sent back, and new phages can be isolated against those resistant isolates in a continuous, sustainable process. So in Belgium, we now have a process for a regulatory framework for allowing the personalized model for phage therapy, where a phage bank filled with characterized phages is stored in a banking system. Those phages are then manufactured in a controlled environment as active pharmaceutical ingredients. So these APIs, each an individual phage, are produced using a suitable host. They're produced according to a monograph. And then external quality testing is performed by the Belgian Institute of Public Health called Cienzano, such that they're then available in hospital pharmacies, where upon a prescription, based on a companion diagnostic, one or more phage APIs can be selected and then mixed in whatever carrier form is necessary for that individual patient, quite flexibly. So this is all governed by a monograph, which is publicly available. It's been published in an open access journal. You can just Google the magistral phage, come up with the paper, and the monograph is in the supplemental information. It's quite short. I really recommend reading it. Where it basically all it does is it defines the concept of the magistral phage. It then defines the manufacturing process that will be necessary. It defines the production environment that manufacturing is necessary to be produced in. It defines the equipment and materials that are necessary to use. It defines the validating tests that must be performed. It defines the host bacteria that must be used. And then it defines the phages themselves that are allowed to be used, which must each individually be approved by the Belgian Institute of Public Health, Cienzano. So it then also defines the storage conditions and how the shelf life is defined, and how the labeling works, as well as how the surveillance works. So this is an example of a phage genetic passport. This is sort of the particularly interesting part, where this has been produced by Cienzano, the Belgian Institute of Public Health, where it just goes over the aspects of the phage identity that are relevant for safety. So particularly the transducing capacity, which is defined by the terminase and the packaging mechanism. It describes the lysogeny-related genes, whether they're present or absent, using a validated assay. It also talks about genes related to antibiotic resistance, insertion sequences, and transposons, more or less exactly what you'd hope would be in here. That would then be relevant for a pharmacist making a decision, pharmacist and physician making a decision about which phages are most appropriate for therapy. So then, this is the assay that determines that this specific phage has a head-full packaging mechanism and thus is unlikely to transduce. But the important part is that it is approved. It's been stamped goedgekeurd, or approved in Dutch. And thus this phage is allowed to be used by pharmacists and physicians in human patients as part of the non-experimental standard of care. But that's just the phage identity. There's then also another report that needs to be generated for each batch, which Cienzano also generates. So this discusses the endotoxin levels, the bioburden, or sterility depending on context, and then the pH of each batch, as well as defining the stability and the abundance of the phages within each API. So this API was thus marked approved, goedgekeurd, allowing it to be used. And this has since been used in patients according to the magistral model. So how it works in practice. phage research labs, or our own lab, generate characterized phages, which then come to us as either licensed or owned phages, which are then banked at the LGM in Belgium, which then sends those phages to the lab MCT at the Queen Astrid, which manufactures them as APIs. And so then the phage and the batches then go to Cienzano for testing, and then come back to us for distribution to the pharmacies. So then they're then available in the pharmacy for a prescription by a medical doctor, who then, using a companion diagnostic, provides the prescription to the pharmacy, who then fulfills the prescription and provides it to a patient to hopefully cure patients, which is the overarching goal of all of this. So we feel that this model has a number of advantages. It's, we think, much more economically and also epidemiologically sustainable than other models. And we also feel that particularly that it allows for a deeper and more open collaboration with academia, where we can feel much more confident in just providing our phages to academics to allow you to hold us accountable, both in terms of the identity of the phages and the safety of what we're doing. And also just all of the various contexts of what we're doing on all of the various levels that you study. And it also allows us to react much more quickly to new threats. So with that, I'd like to thank our team at Vizal, and also the team at the Queen Astrid Military Hospital, who pioneered this entire model. Jean-Paul Pierney will be giving a talk discussing the specific experience at the Queen Astrid later this week. So with that, does anyone have any questions? Susan.
Wait.
How long does it take for you to identify a phage specific for a specific isolates, for the patient's isolate?
So we do this using standard techniques with a few extra steps that we feel adds to the efficacy of our phages, using directed evolution techniques. But we can, this is depending on the strain, on the order of a week to weeks.
So what is your product here? Is a phage library, the whole library is the product, or the single phage is the product? What is your whole product?
So the product is both both access to the phage library, the phages within the library, and the companion diagnostic.
Okay, thank you.
Hi, I'm wondering if you can elaborate a little bit on the companion diagnostics.
So we are currently developing a robot that will be able to perform this diagnostic on single culture isolates within 2 to 3 hours. So it will fit into the schemes that clinicians currently have for an antibiogram. It will function largely like an antibiogram.
All right, Bob, I'm going to disagree with you that fixed cocktails and patient-specific cocktails are two models that are in competition with each other. So they have different strengths.
Absolutely.
I think it's absolutely fair to say they have different strengths. I'm very wary of framing it as two things in opposition.
Absolutely. They do very much both support each other, where a lot of the evidence that has supported cocktail-based models has been generated using the personalized model, and a lot of the techniques that are currently supporting the personalized model were developed using the cocktail-based model.
So one of the big questions I have for the model that you're describing has to do with essentially what you have the opportunity to test, because you can obviously do a lot of characterization and quality control on the individual phages. What isn't clear to me that is part of that, and this is sort of, I'm very curious as to how you address this next bit, is what happens when you combine them? A physician and a pharmacy decide that phages A, B, and C are the ones that are helpful, and presumably they have information about them to know that these are different things, they can do different things, but you may have never tested that combination to find out if, the three of them together don't have stability. And I know that in mixing immediately patient bedside model, long-term stability isn't quite so much a concern that exists in that model, but even short-term stability can be surprising sometimes, especially since you mentioned putting them into a hydrogel or, and you didn't say bandages, but I think like that's the kind of thing that's been discussed. Those are all other substances that are now present, and unless you've tested a lot of different combinations of your phages with a lot of different combinations of these different delivery mechanisms, you could have something very unexpected happen.
Absolutely. This is very much something that we've considered. The stability that is relevant to this model is on the order of days, which reduces the amount of trouble that we can get into, but it doesn't totally eliminate it, absolutely. And so once we've assembled our complete library for each of the bacteria that we're going to be treating, we're going to mix all of the phages together and assess the stability of each of the phages individually within that mixture in order to identify the stability of each phage individually in combination with all of the other phages that it could possibly be mixed with. Yes, Evelyn.
Hi, Bob. I was wondering, how scalable do you think this model is to a country that's not tiny and has only 10 million or 11 million people?
So the model is already tentatively scaling to France, where we've gotten interest from the competent authorities there, and the Magistral model has already been applied in France already. But really we see this scaling not to countries but to clinics. So to phage therapy centers, which as soon as we gain approval from competent authorities in a member state, we're at the moment only really thinking about Europe, then clinics will be able to join our family.
So that sounds awesome for Belgium and France, which have a completely, let's say, socialized compared to the US, healthcare system. How would that translate into the US and its healthcare system?
So I think this model would be difficult to translate to the US system, particularly because the equivalent model for regulating this kind of therapy, the rules are much more lax in the United States, and perhaps the rules for compounding therapeutics are not appropriate for phage therapy. This is a conversation that we're going to need to have with Scott at some point in a few years. Where we'd want to be regulated more than the current rules really allow.
But it's not just about but it's not just about the regulation. It's also about, especially in a model of a company like yours, where you're working on profit based on your phage and your phage magistral preparation, including those diagnostics. How would that translate into a company here, or any kind of pharmaceutical being interested in?
Well, I think this would be a better question for API, APT, which is attempting to create this model in the United States, and they have more experience thinking about the American market. Yeah, at some point I'd love to hear your thoughts on this.
Okay, thank you.
Thank you.